Okay. So my name is Yi. I'm from the bioengineering department. So the research focus of my new lab is on the fusion oncoprotein-driven sarcomas. So the sarcomas are quite different from most other common cancers that you may have heard about, like colon cancer or lung cancer, in at least two different ways. Number one, you can see here, instead of skin or epithelial cells, sarcomas originate from connective tissues such as bones and muscles. And even more importantly, sarcomas compared to many other more common cancers often are driven by the fusion oncoproteins. I know many of you are from the computer science background, and you write a lot of code, and sometimes, unfortunately, you will make mistakes. So you call it bugs, right? So in the sense or in the context of human genome, we call it genetic mutations. So when cells divide, unavoidably, they also make mistakes or those bugs. So common cancers like lung cancer or breast cancer often involve dozens, if not hundreds of such mistakes. Then in contrast, these fusion-driven sarcomas often have surprisingly very low number of mutations, sometimes down to just one mutation, or basically just one mistake. As you can see from the panel A, for specific fusion-driven sarcoma that I have been working with the other two professors in the last 5 years, is a NAPSTAT-driven SFT. So what's happening is a piece of genetic materials or information get misplaced in other places of human genome. The end result is two genes get fused together, and that become an oncogene, which means that it can drive the formation of cancer. So what my lab is planning to do, we will focus on a specific group of such fusion oncoprotein called EWS1 fusion-driven sarcomas. As you can see here, what we are trying to do first to use different genome engineering tools, such as CRISPR, to build this kind of sarcoma models. And then using those models, then we will study the mechanism. What that means is we are trying to understand why this misplacement, this bug, can start the cancer formation or progression. And then, of course, one of the focuses is therapy. So we will try many different ways, such as more traditional drug screening or RNA therapeutics, and especially the cancer immunotherapy. Maybe some of you already heard about. You can see from the panel C. So what we hypothesize is these fusion-driven sarcomas may express specific fusion-derived peptide on the cancer cell surface. So they will be the ideal target for the treatment. Yeah, thank you.